click on the links. And we're hanging out here, waiting for things to go live, because that's what we do. Oh, hey, I think we're live. Hello, everybody. 3, 2. This is TWIS. This Week in Science, episode number 639, recorded on Wednesday, October 4th, 2017. Science's all-male review. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your heads with raccoons, old people, and herpes. But first...
Disclaimer, disclaimer, disclaimer. We here at TWIS headquarters understand that sciencey news may not be for everyone. Stories here typically begin with phrases like, recent research into fill-in-the-blank reveals, or according to new fMRI study on insert cephalopod here, or unearthed fossil found of previously unknown fill-in thing you never heard of existing before. Or even an interesting discovery from NASA, just beyond the insert somewhere in space. Only some people in our society want to hear stories like these, so we don't do the show for everyone. We do this just for you, because you are one of us. One of us who finds discoveries in science not just informative, but exciting and inspiring. One of us who revel in knowing, thirst for more data, hunger after every study for further research to be done. And while the news world at large continues to pander to the popular pulp, keeping science news far off in people's peripheral, you are one of us here to fill in the blanks of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back once again with all of our favorite stories from the week to talk about them, to opinionate on them, to really get into the science, right?
Yeah, yeah.
We love it. We love it. And we're so glad that you're here with us. So thank you for being here. On tonight's show, I have stories about Nobel Prizes, the actual Nobel Prizes. And
I didn't check my mail.
Yeah. It would be a phone call. You would have gotten a phone call. It's not just yeah. Anyway, CRISPR and clones. That's right. The clones are being edited, and a Tabby star update. Justin, what do you have?
I've got modern humans older than we think, and evidence of Italian Celts. Huh?
That just doesn't even make sense.
Yeah, not yet.
Okay.
Not yet. It might never.
And that's it. Okay. And then moving on, Blair, what is in the animal corner this week?
Oh, I have the aforementioned trash pandas. I have freeloading invertebrates. And I have
a British accent.
Killer scientists.
Killer scientists?
Yes.
Oh my goodness. Is this going to be one of those new Science Channel documentaries?
Yeah, it's... Little did you know, the scientists lurking around the corner.
That's right. Okay. I can't wait to get into all these stories. It sounds like a lot of fun. But before we do, we have to jump into our new favorite segment of the show. And I want to remind everybody also that you can subscribe to the TWIS podcast if you are not already in just about every RSS feeder that's out there. iTunes, the Google Play podcast portal, Stitcher, Spreaker, TuneIn. Just look for This Week in Science. You can also find us on YouTube and Facebook if you want to watch us while we do this thing that we do. Again, look for This Week in Science. You can also find links to everything and most recent episodes at twis.org, where also you can find a way to pre-order your annual Blair's Animal Corner TWIS calendar. They're available now. So if you go to twis.org, you can pre-order that right now. But here it goes. We're ready for This Week in What Has Science Done for Me Lately? Tom McCaig writes in to say, in 2012, I was diagnosed with melanoma. Even with treatment by early 2014, it had metastasized and the prognosis was bad. After 4 treatments of Yarvoy, followed by 36 biweekly treatments of Opdivo, I have had a complete response. No signs of the cancer for 10 months. That's what science has done for me lately.
That is awesome.
It is.
Can't really beat that.
And we're glad that you're still with us and that you're here to write in and let us know how the treatments have been working for you. And thanks, science, because seriously, science helps us keep people around. And that's just awesome.
Happy to have you with us, Tom.
Very much. Thank you so much for writing in with your story. Anyone out there who wants to write in, remember that you can leave us a message on our Facebook page. So just go look for This Week in Science on Facebook, facebook.com/thisweekinscience. Write in with your stories of what science has done for you lately. I know they are many and varied, and we want to hear from you, and we want to read them every single week. So keep them coming, you guys. I'm loving this. Is it time for science now? Are we gonna discuss this science thing that we love so much? All right, so let's do the celebrating first. Let's get the celebrations out of the way so we can get into the other things. I would like to congratulate Science's all-male review. And by that, I mean the Nobel Prize winners this year. Every single Nobel Prize recipient for science is a man.
You know what, Kiki?
And a white man at that.
Finally, white men in science are getting their day in the sun. Am I right?
You are right. I mean, they are. It's great. And I'm not going to malign the discoveries that are being celebrated this year.
Just the gender and race of those who are making them.
Yes, absolutely. So the Nobel Prize in Chemistry for 2017 has been awarded to Jacques Dubochet, Joachim Frank, and Richard Henderson for their development of a methodology using cryo-electron microscopy to freeze biological molecules mid-stride, to be able to portray them in action at atomic resolution so that we can get a picture of what the things that make up our cells and that make up the things inside our cell, the things inside our cells actually look like and how they work. We have gotten to see ion channels in action as a result of cryo-electron microscopy. It took biochemistry to places it never was able to go before. We have an inside view of the structure of potential targets for drug treatments. We have an inside view of the lock and key mechanisms that are in place in our ribosomes. Like, there's so much that electron microscopy and this methodology has allowed us to do. It really is deserving of this celebration. The researchers, this is from the nobelprize.org website, Jacques Dubochet says, explains how the technique of water vitrification that he came up with has allowed us to observe biomolecules like Zika and Salmonella in their natural state. He says, if you have them in ice, the molecules, like fish, are dead. Joachim Frank says, normally my dog wakes me early in the morning, but today it was the Nobel Prize on how it felt to be receiving the Stockholm phone call.
Must be nice.
Yeah, and Richard Henderson says it's opened up a previously unapproachable area of structural biology on his part in the discovery. The prize in physics for 2017, it really isn't a secret. We've been talking about the 4 discoveries that have been made over the last couple of years in great detail. It was something that on this show over a decade, we debated as to whether or not they would be discovered. But this award in physics this year has gone to Rainer Weiss and the other half jointly to Barry C. Barish and Kip S. Thorne for their decisive contributions to the LIGO detector and the observation of gravitational waves. Rainer Weiss, he came up with the idea back in the 1970s originally, and he explains why measuring the effect of gravitational waves is so very hard to achieve. He says, space is enormously stiff. You can't squish it. Barry C. Barish talked about the LIGO detector and the size of the actual signal that they detected the very first time. He says the actual size of the signal was about 1,000th the size of a proton. 1,000th.
That's not even a measurement. What is that?
It's less. 1,000th of a proton wiggled, and this detector detected it. And we went, black holes have merged. We know things now. It's the scale of this is mind-boggling. Kip S. Thorne says, huge discoveries are really the result of giant collaborations. And this is a point that many people have been bringing up recently, and a point that, again, I was kind of alluding to as with the all-male review joke at the beginning of discussing this story, is that really big experiments like the gravitational wave detector, LIGO and Virgo together, the papers that are published on these discoveries, the first paper had hundreds of contributors. You know, these are huge team efforts. And in chemistry as well, all of these discoveries are step-by-step team efforts. It's not one person, it's everyone in the lab. You look at any paper, there are multiple authors on most papers that are published. The primary author, the last author, is the one who usually they're the head of the lab, the one who got the funding to make it happen. They've been working probably the longest. And so they get the spoils of history. But science really is a result of these collaborations. And if not a collaboration, then multiple discoveries occurring in a fashion that allows understanding to take place. The final award I wanted to talk about tonight is the prize for physiology and medicine. This was awarded to Jeffrey C. Hall, Michael Rosbash, and Michael W. Young for their discoveries of molecular mechanisms controlling the circadian rhythm. And so Jeffrey C. Hall praised the role of the fruit fly. And he says the key 4th awardee here is the little fly. Michael Young said that he took on the mystery of circadian rhythms because he just thought it was a terrific problem. And then Michael Rosbash says when he was awakened by the Stockholm telephone call, he says, my wife said, start breathing. This one is particularly interesting because it was the discovery of multiple clockwork mechanisms in cells of the fruit fly. Initially, the clock that was discovered, there were genes that were discovered that they named period, and then a second gene group that was called, let's see if I can find it here, that was called TIM, or TIM, the period gene, and timeless. It's abbreviated TIM. So period and timeless work together in a cyclic manner to give us our 24-hour cycles. And not just us. This has been discovered in plants, in yeast, in worms,
Fruit flies?
across this, fruit flies, across the spectrum of life. And many other discoveries have come out of this as a result. These genes regulate everything about life.
Where is the teeny tiny Nobel Prize for the great, 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 great grandson of the fruit fly in the study?
Right. Yes. So no women won an award this year.
This year. There's always next year.
Yes. And according to the Bad Astronomy blog over on Sci-Fi Wire, Phil Plait, he says that the nomination list for this prize is embargoed for 50 years. So we can only look at data up to 1966. And if you look at the list up to 1966, there were around 2,500, well over 2,500 nominations to men and only 98 to women. So until 1966, women were barely nominated for any of these awards. I'm going to suggest that the last 50 years is going to show an improvement in that. I'm going to suggest that the last 50 years has shown an increase in involvement of women in science and publishing crucial papers to research. And I'm going to suggest that some 50 years from now, we will see that this year there were many women nominated. However, the discoveries that they decided to award this year were these very important discoveries that just happened to be awarded to men. And I predict that as we move forward through the next 50 years, many more Nobel Prizes will be awarded to women and to people of color. The diversity is going to go up because diversity in science is going up.
Yep.
It has no place to go but up and to get better. So.
Also true.
Yes.
Well, yeah. And I mean, I think I've done this before, but I've sort of put this in the light of something like Title IX, where if you're going to talk about adult athletes who are competing, even you look at the Olympics is a great example. Before Title IX, there weren't very many female Olympians, and it took a generation of women who were doing sports and starting in schools early on for there to be enough female athletes in this country to be competing in the Olympics. And I think the last time around, the women brought back a few more medals than the men. So it's just sort of one of those things. Women were for a long time sort of locked out of academia at the higher levels. We're talking about this one study where this whole thing, that the proof of this has been discovered, and the initial hypothesis was in 1977. So this is a generation long project that's being awarded, and it's coming from a time back when there weren't as many women in science with access to this. So the idea that it's in the pipeline, and that's exactly right, Kiki.
It's in the pipeline. Yep.
And as Bleak says in the chat room, tune into TWIS in 50 years to find out.
That's right. And okay, this has been some awesome celebratory science. It's wonderful that we have made such discoveries. These discoveries will be the shoulders that future discoveries stand upon. But now let's talk about clone editing.
Okay.
Yeah, the Chinese are at it again. They were one of the first teams to report using gene editing, using the CRISPR-Cas9 technology to edit genetic mutations that were deleterious and hereditary, to use early embryos, to use CRISPR-Cas9 to go in and cut out bad genes and to try and repair these deleterious mutations. It kind of worked, but it kind of didn't. And since then, we have Shukrat Metalipov here at OHSU in Oregon, who has shown His methodology of using the CRISPR editing technique on early embryos was very successful, potentially, and it's still a little bit controversial and under debate in the scientific community. But what else is science for than healthy debate? But the Chinese have published a new study in the journal Protein and Cell. Not only have they used gene editing, they have basically created clones to do it. They wanted to fix a recessive hereditary disorder. So if you know anything about the way genetics works, dominant mutations only need one copy of the mutation to be expressed and to show themselves. Recessive traits, however, they need two copies, one copy on each chromosome to actually show themselves in the individual. To do that, though, it's very hard to find individuals for their research that actually had these double recessive mutations. To get around that, they developed embryonic clones from some patients' skin cells. They found people who had this hereditary disorder called beta thalassemia. It's a blood disorder and can be really terrible. And they had some patients use their skin cells, created clones of these individuals. So all the cells would basically be the exact same as the original individuals, created these little early embryos. They don't let them grow past 14 days. And then they used an updated version of CRISPR that goes in, and instead of cutting out a gene, it goes in and clips out just one little base pair. So we know DNA, RNAs made up of guanine, adenine, cytosine, thymine, right? So, and uracil. But instead of taking the whole gene that's like ATT, AGCC, whatever that combination of letters is, it goes in and looks for the one specific mutation, the point mutation that has gone wrong, that has changed the gene and the protein. Goes in, fixes that one little thing, and they actually had reasonable success. It wasn't great. 8 out of 20 cloned embryos, they were able to convert a guanine that wasn't supposed to be there, into an adenine in either one or both copies of the gene. And in doing that would make this recessive trait not be expressed. One of the issues with this, though, as well, is that they did have a problem with mosaics, which means these individuals, these little embryos, these little balls of cells, not every cell in the early embryos took up the editing in the same way. And so some of them got edited, some cells had both copies edited, some cells had one copy edited, and so depending on the cell, you may or may not have expression of this deleterious trait. So this isn't a slam dunk for the Chinese for sure, but it's a very interesting potential for future avenues for therapy of this disorder, beta thalassemia, and other inherited diseases. And it does show this this interesting fixing of point mutations. Most human disorders are caused by point mutations and not big gene problems. Most of them are just single point mutations. And so this technique, this is a proof of concept that they might be looking in the right direction. Yeah, so it's pretty exciting. The Chinese are going for it. It's very exciting. Very exciting. We'll see. The beta thalassemia is actually a recessive disorder that is a big problem in China. And so it's a very important disorder for them to go after at this point in time. But you know what? Let's not talk about this point in time. Let's take it back.
Okay.
Back in the time machine. To when we were chimpanzees. And we had herpes. We had herpes when we were chimpanzees.
Well, we
right?
were never chimpanzees.
We weren't.
We just
Before we
shared a
split.
common
An
ancestor with
ancestor.
chimpanzees. Yes.
Thank you for clarification. Yes. Anyway, our common ancestor, the common ancestor we had with chimpanzees, had herpes, cold sores, herpes simplex virus 1, and genital herpes, herpes simplex virus 2. We split off from that early ancestor. The chimpanzees got lucky and they kept on carrying both types of herpes, type 1 and type 2. They held on to both of them.
That's the lucky part.
But our ancestor that split off, we only took herpes simplex virus 1 with us. We lost the genital herpes for a while. There's something happened, right? This was about 7 million years ago. But then about 3 to 1.5 million years ago, genital herpes came back. We got it back. And so these researchers from Cambridge and Oxford, publishing in Virus Evolution this week, I think they looked back in time to figure out how it jumped back. Where did the genital herpes come from? We lost it, and then we got it back. And how did that happen? Basically, they think that there was a bipedal hominin called Paranthropus boisei.
Oh, Boise's.
Boise's. That's
Big
right.
Boise's.
Yeah, Paranthropus boisei scavenged chimp meat. It was a scavenger. Probably ate chimp meat that was infected with herpes simplex virus 2, thus probably contracting.
That's how they're telling it anyway.
Themselves, yes. It could have been through open sores that the meat may have, or the blood from this carcass may have gotten into some kind of infection, an open sore that the individual had. It's probably happened multiple times at this point in time. And then there was close contact between boisei and Homo erectus. And it was at that point that boisei gave genital herpes back to Homo erectus.
By way of a hug.
By way of a hug.
This is why you need to have a conversation with your partner, even if they're a different species, about their sexual history. It's just important.
It is important. Yes. Researchers from, there's some great comments from the researchers. Senior author Dr. Charlotte Holdcroft from Cambridge's Department of Archaeology says, herpes infect everything from humans to coral, with each species having its own specific set of viruses. For these viruses to jump species barriers, they need a lucky genetic mutation combined with significant fluid exchange. In the case of early hominins, this means through consumption or intercourse, or possibly
Wouldn't rule
both.
it out.
Could be both. Could have been both. I mean, it could have been also that Homo erectus, being a scavenger, scavenged dead, infected Paranthropus boisei carcasses as well. I mean, this could have been a very interesting chain of jumping.
Herpes is crazy.
But we don't have.
There's another kind of herpes that when you work with primates, you have to be really careful about. It's called herpes B. And that stuff they carry, it doesn't really affect them, but just from like a splash into your eyeball or something like that, it can get into your body and it can basically eat your brain and you die in like 48 hours.
I don't want that.
No.
Yeah.
Nope.
No.
That's terrifying.
Herpes is a scary thing.
Oh my goodness, terrifying. Yes, and additionally, Holdcroft goes on to say, once herpes simplex virus 2 gains entry to a species, it stays, easily transferred from mother to baby, as well as through blood, saliva, and sex. Herpes simplex virus 2 is ideally suited to low-density populations. The genital herpes virus would have crept across Africa the way it creeps down nerve endings in our sex organs, slowly but surely. Yeah, so this team is going to their next step. You always wonder what the next step is, right? They are
What's next?
looking at climate change and genetics and timing of things. They were able to tease out this story, but they think they can do the same thing to figure out things like where humans got pubic lice. That's their next. Yes.
Because obviously that's natural progression.
It's a mystery of an ancient disease.
Really? We don't know where it came from.
A modern yet ancient disease.
Yeah, because we call it crabs, but it's actually a lice.
That is very
Not actually
specific.
from the ocean.
It is an organism that is very specific to its environment. Pubic lice they're not the same as head lice.
Right.
They are different species. So
but we have them, I think, in common with gorillas.
Yes, and so it's thought that pubic lice were introduced similarly to the herpes simplex virus 2 via an intermediate hominin from ancestral gorillas over 3 million years ago.
I wonder if somebody was wearing gorilla skin or something like that.
No, look, you stop. Just quit trying to come up with different ways that this could have happened. And let me tell you, it wasn't 3 million years ago either. Okay, look, one thing that we have been learning about early hominin history is that they had sex with each other a lot, and as we know, not every species that's a little bit distinct can mate and reproduce, which means they were mating even when reproduction wasn't taking place. We have plenty of examples where reproduction did take place in different degrees. That does not mean they stopped having sex when, if we can't have kids, then forget it, right? No, they were all hominins have always been having sex with each other all the time. Yeah. And even those that like Borealis, that Borealis is kind of more on the ape side than anything that we would consider human.
Yes.
Right? Kind of gorilla-y. So there was a time when, gorilla, Homo sapiens, it wasn't that far away really yet. It all looked good, and that's what happened. There was a lot of hominin sex taking place. That's
it's like a game of telephone, but a little bit different.
That's why we don't have. Yeah, that's why they don't call it like the direct ancestral links going back. They now call it a braided stream, which means lots of disparate hominins were having sex, and some were successful in reproducing, which would then go on to reproduce. So you can't tell what's really this from that at some point. And that's how we ended up with people, because of sex.
It is.
I mean, what a moral to the story.
I mean, you can't take that away from us, right? This is how
where do people come from?
Sex. That's the basic answer. Yeah. And speaking of which, this is This Week in Science. Justin, let's move into your story, which I think is pretty related to this.
Yeah, it says here, modern humans are a lot older than we think. This is an amazing discovery. Discovery of a 2,000-year-old South African child. Wow! And while that story would be even more interesting if we had, in fact, discovered a child that was alive and 2,000 years old, the remains of this child who lived 2,000 years ago, before there was much migration from northern to southern Africa, he was a South African child after all, gives us a decent genetic clock to gauge the emergence of modern man. Based on the findings, we may be about 100,000 years older than we had believed. This is Marlies Lombard, professor of the Stone Age archaeology at the University of Johannesburg, initiated collaboration with geneticists from Uppsala University in Sweden and the University of Witwatersrand, who put together a team of experts at Uppsala Laboratory. They reconstructed the full genome of the Ballito Bay child, together with the genomes of 6 other individuals from the KwaZulu Natal who lived between 2,300 and 300 years ago. 3 Stone Age individuals who lived between 23 and 1,800 years ago were found to be genetically related to descendants of the Khoisan group living in southern Africa today. So very much the same direct ancestry. The remains of other 4 individuals who lived 500 to 300 years ago during the Iron Age were genetically related more to present-day South Africans of West African descent. Because the boy from Ballito was of hunter-gatherer descent, living at a time before migrants further north in Africa reached southern shores, his DNA could be used to estimate the split between modern humans and earlier human groups as occurring between 350 and 260,000 years ago.
Okay,
This means-
I'm missing something. I don't
Yes.
understand how you get from a 2,000-year-old remains of a human to a 200,000-year-old estimation of the emergence of man.
Right. So you have these. One of the sort of wonderful things about Africa is that it's really big, and the groups of people there going back in time were relatively small. So you have very isolated groups who were isolated for a very long time. And what they were doing is comparing the genetic changes over time in this one child to those of disparate groups from elsewhere, and they could backtrack sort of genetic. I guess the point in the going back in the family branch where those two were of one ancestor. Right? And that number came out to be between 260 and 350,000 years ago of two disparate groups of modern humans, right?
Wow.
Now,
Yeah.
that estimate coincides with the Floresbad skull, who was a contemporary of Homo naledi, that relatively recent addition to the hominid family. So according to this, it now seems that at least 2 or 3 Homo species occupied southern African landscape during this time, which also represents the early phases of the Middle Stone Age, says Lombard, one of the researchers involved. Pulling it all together, the fossil, ancient DNA, and archaeological records of Africa indicate that the transition from archaic to modern humans might not have occurred in only one place in Africa. Instead, regions including southern and northern Africa probably played a significant role. So it's also, I guess, it sort of points out because the northern Africans and southern Africans sort of diverged that long ago, 350, 250,000 years ago. It also might indicate that the emergence of Homo sapiens was occurring in multiple spots around Africa at that time as well.
I think it's just amazing to think about all the different, I mean, we used to think it was almost this straight line, right? And that didn't make sense. But now we're actually finding the fossil records. We're finding the genetic evidence to support, like you said, this braided rope, these multiple lineages, these different groups of hominids. I think it's just fantastic. And it goes back longer than we thought previously.
Yeah, the idea
We're
that
old. all this advancement, our development into who we are as humans, Homo sapiens. These things, it's got an earlier start. The further back we push it, the more sense it starts to make.
Yeah. Of course, even though that's pretty dramatically far back, there were still already Neanderthals in Europe and in Siberia, even up through. I mean, Neanderthals were already roaming around all of Europe and much of Asia probably as well. So, and then Denisovans, and then there's people everywhere.
There are people everywhere. It's great. People everywhere, all over the planet. And now we're one people because of sex. That's what we learned.
Because we had so much sex, it turned all those disparate groups into one.
I think we have to change the title of tonight's episode to Because Sex.
And on that note, everyone, do you know what time it is? It's time for Blair's Animal Corner.
Ah.
I was going to say Blair's Invertebrate Sex Corner.
I don't have that, though.
She loves our creature, great and small. Biped, millipede, no pet at all. Wanna hear
hear about animals. She's your girl, except for giant pandas and squirrels. That I'm
What you got, Blair?
I wish I'd gotten the memo. I don't have any invertebrate sex, but I do have the story of the crow and the pitcher. Are you familiar with Aesop's fable of the crow and the pitcher?
Oh, yeah.
Yes. So there's a thirsty crow, comes upon a pitcher with water at the bottom, but it's too far down. It's beyond the reach of his beak. So he tries to push it over, can't do it. He then drops pebbles in one by one until the water rises to the top because of displacement, right? And the morals there are that, let's see, thoughtfulness is better than brute strength, that necessity is the mother of invention, and that where there's a will, there's a way. Well,
Aesop just probably saw a crow do that and was like, oh, this is cool.
Exactly. So this is something that actually crows have been doing for a long time, and Aesop most likely did. That is the kind of the hypothesis here, is that Aesop watched this happen, and that this is kind of just it was an example of animal ingenuity. Right.
And why haven't crows ever gotten the Nobel Prize?
Great question.
This was a study or an observational study that I did. I was just describing. I'm not.
Yeah. In fact, I did a little research. I found that some evidence that perhaps Pliny the Elder is the earliest to find this behavior in real life corvids. But we're not here to talk about crows because we know they're smart. We've talked about them a million times on this show. Instead, I want to talk about raccoons.
Raccoons.
Trash pandas.
Trash pandas, raccoons.
Yeah.
Yes, so a group of U.S. scientists led by the University of Wyoming have decided to do this pitcher test on raccoons. So first, disclaimer, 8 raccoons, all in captivity, were the ones for this experiment. So definitely, no matter what I say next, more study is needed. Yes. So the research team, they wanted to see if raccoons would be able to drop stones into clear tubes of water to retrieve floating marshmallows. They found that the raccoons, similar to studies of non-corvid birds that they've done before, did not just walk in and immediately go, oh, this is how to do this, and drop stones into the tube. Instead, they had to kind of train the raccoons how to do this. They did that by balancing the stones on the rim of the tube, and raccoons, as they kind of walked around, would accidentally knock one in and then go, oh, the water went up. Maybe I should do that again. And then they would do it again and again until they could get to the marshmallows. So then that taught them stones falling into the tube, brought the marshmallow closer. So that's definitely, it's a different level of ingenuity than they were hoping for. During the training, 7 of the 8 interacted with the stones. 4 raccoons retrieved the marshmallows after accidentally knocking stones into the water. 2 of the 4 got the marshmallow and then learned on their own to pick the stones up off the ground and drop them into the water to get a reward. So only 2 out of the initial 8 then, after accidentally doing this, decided, oh, next time I'm going to go ahead and pick up this stone over here and put it in the water. So only 2 of the 8. And then a 3rd raccoon surprised the scientists by inventing an entirely new method for solving the problem. She found a way, despite their attempt to weigh it down, to overturn the entire heavy tube to get to the marshmallows, which is classic raccoon, in my opinion.
I'm not going to do your study. I'm going to just do what I want. Trash panda it.
Look, there's a very complicated looking latch on top of this. I think if I take a running start from the roof, the eave of the roof, I could land on this thing and just pummel it down the road and break it open. Let me do it that way.
Yeah. So then the other kind of part of this experiment, which kind of didn't have any conclusive results, was that they wanted to have different objects that the raccoons could drop in, things that float versus things that sink, bigger things versus smaller things, heavier things versus lighter things, because it's all going to displace water differently. And they didn't see anything too interesting or conclusive in that, except that they performed differently than birds or human children did in this trial. So they couldn't conclusively say whether the raccoons understood, if they had problem-solving abilities from this, but they did say that they acted very differently than birds and humans. So they found that raccoons could be innovative in many parts of the task, but that they had fewer successful experiments with it. But they also do say that the raccoons had fewer opportunities and fewer kind of different variables than in previous studies. So they want to do more, of course. And they think that if the raccoons familiarized themselves with it, with the stones, the tube, the whole setup, that it would come a lot quicker to them after familiarization. If you think about a raccoon in your trash can, no matter how many different ways you try to kind of latch down that lid, they will find a way to open up your trash can and get to your trash. That's because they're very familiar with that apparatus, right? So they say that their study demonstrates captive raccoons are able to solve novel problems and that they approach classic tests of animal cognition in diverse and exciting ways. They can't wait to see what they do next. Yeah. So all in all, not super conclusive of a study, other than to say that they have a very different way of going about solving problems than some of the other animals that we've studied in these kinds of problem-solving experiments previously, which makes sense because they're urbanized animals. They are in a really specific kind of environment where problem-solving, it's hard for me to believe that they don't have intense problem-solving abilities, because
Absolutely.
you need to live in the big city. Right.
Yeah, not only do they live in an urban environment, beyond that, they're generalists and they are social animals. They live in groups. They usually, the parents are very closely involved with raising their young. So there's probably some amount of skill transfer that goes on between generations. So you've got generational inheritance of skills. And then they're generalists like rats and crows and pigeons. These animals succeed. Raccoons are succeeding in the urban environment because they're able to solve problems.
Right. But one of the things that the lead researchers did mention that I will just say kind of as a last thought is to try to parse out the difference between intelligence and problem solving and exploratory behavior with extremely dexterous paws, which is a good point.
So I've been trying to teach my cats how to use one of my cats likes to scarf and barf. So I'm trying to stop that from happening.
Right.
And so I've bought this little ball device that I can put the food into and then open up so little holes are available, and then teach the cats that if they knock the ball around, little bits of food fall out, and then they can feed themselves and play at the same time. Sounds great. One of my cats has figured it out. The other cat refuses. The first cat is more exploratory and is bolder, is more of an outgoing personality type cat. And so I wonder if the personality types have played into their exploratory behavior and their each exclusive ability to understand and do this task that I've set for them.
Absolutely. Yeah. And I would argue that is one of the reasons that I respect and not fear, but definitely am wary of raccoons when I come across one at maybe 4:30 or 5 o'clock in the morning on my street. It's because of the boldness. And that exactly is the thing that that can make raccoons extremely successful, despite potentially a lack of intelligence or problem solving, which is what this whole. So that's kind of the question, right?
So maybe raccoons are just bold and stupid.
So
Just
it's
persist.
not even that. It's maybe they're much more likely to pick up strange things and put them down in new places.
Yeah.
Right? So, which is a good question. Is if you're an animal with something akin to thumbs, not quite thumbs, but if you're able to pick things up really easily, move them around, push them down, push them into things, if you're more dexterous and you're also not afraid, are you more likely to find solutions to problems, stumble across them, perhaps by chance?
And
That's
and
an
it
interesting question.
Mhm. Increases
It's about-
probabilities. Yeah.
also a statistic I just made up on the spot. 8 out of 10 attic hauntings are actually raccoons.
Yeah, probably.
The other
Are you
two,
sure? The other two?
the other two are pigeons and or bats.
Ah.
Most likely possums.
Mhm.
Could be possums also.
Yeah, possums
Yeah,
are in
yeah.
there too.
None of them are giant spiders. Okay, so
There we go. Moving on
moving
from raccoons,
on.
not talking about giant spiders. And
Nope.
now what do you have?
Now I want to talk about tsunamis. So tsunamis are front of mind today and lately. Tsunamis are devastating to humans and human spaces affected by them, but they're also a phenomenon that requires study because they change things drastically very quickly. And I wanted to highlight a recent study that was looking at a side of tsunamis that perhaps has not gotten a whole lot of attention previously. And that's looking actually at the 2011 tsunami from Japan and stowaways that move because of that tsunami. And what I mean by that are invertebrates hitching a ride on debris and crossing the Pacific Ocean.
That's a long trip.
Finally, we can find a silver lining for plastics in the ocean.
Interesting you say that, Justin. What are stowaways but another word for invasive species? Right.
Yet another reason why plastics in the ocean are bad.
Bam. Okay. So nearly 300 species have appeared on the shores of Hawaii and the U.S. West Coast attached to tsunami debris, according to biologists from the Smithsonian Environmental Research Center at Williams College. They looked at a bunch of different debris that has washed up from 2012 to 2017, and they found that there were about 289 living species on tsunami debris originating from Japan. There are far more that are that escaped their notice in their survey, and while arrivals have slowed down, they have still not yet stopped. The team is still finding new species when the study period ended in 2017. So the interesting thing is that most of these coastal organisms, they can't survive at open sea. So they normally could not survive this long 1 to 6 year journey across the Pacific Ocean. This is a new opportunity because of plastic, because of storm debris like docks and ships. They create a very slow moving but stable platform for these things to move on. Mollusks like mussels were the most frequently found of all invertebrate groups. Worms, hydroids, crustaceans, bryozoans, these are all animals that they found. 2/3 of the species had never been seen on the U.S. West Coast before. None of the species were known or expected to survive transoceanic rafting voyages. Then that's because the open ocean is really harsh, of course. But these ocean rafts were only going 1 or 2 knots. To kind of put that in perspective for us landlubbers, that's 20 or more knots for commercial ships. So that's really, really slow, just 1 or 2 knots. This means that they gradually could adjust to their new environments, and they actually think that animals were able to reproduce and host larvae on these little floating islands. So
this is oceanic panspermia.
Yes, absolutely. Yeah, so there's a huge potential for marine debris, as Justin was getting to already, to increase the probability of these sorts of things happening. And This has turned out, says co-author John Chapman of Oregon State University, to be one of the biggest unplanned natural experiments in marine biology, perhaps in history. Yeah, scientists largely agree that prevention is the most effective way to combat invasive species, but we can't prevent tsunamis. So the main focus should be on managing plastic, these vessels that these invasives are moving on. Yes, just another reason to ditch plastic.
Plastic, trash, things that do not break down.
Yes, they are.
Sun and salt water over a long voyage.
They are harboring biological criminals.
Well, it can wreak havoc on local coastal ecosystems that are probably already suffering from multiple.
They're like the cats of the ocean.
Okay. But to tell you how a couple of these things might affect, just very quickly, a lot of these animals that I talked about before are filter feeders. So one of the huge differences they can make is if a mussel population explodes, water clarity improves, which at first sounds maybe like a good thing. Not for animals that have adapted to a foggy ocean. So here on the California coast, the water is very cold and it's full of lots of nutrients. It's full of lots of phyto and zooplankton, and it's also full of a lot of dirt and silt. And so a lot of animals depend on those zooplankton, phytoplankton to eat, but they also depend on the murky water to hide from predators. So when you improve water clarity, sometimes that can actually it can destroy ecosystems. Yeah, absolutely.
Fascinating.
Yeah. So it's a delicate balance.
It is.
Yeah.
Such a delicate balance.
It's like when it's 80 degrees in San Francisco and they put out a warm weather alert.
Yes. Yes. That was great.
For the delicate flowers. That's right.
Yeah.
All right, everybody. It is time for us to take a break. We are going to take a quick break and then return with a lot more science. We have a lot of good stories coming up in the next half of the show. We've got Tabby Star being updated. We've got early Italian Celtics from Justin. I don't know. Got a lot of good stuff. I hope that you stick with us. Just a few more minutes. This is This Week in Science.
Explain the things you heard with more than intuition. The line of reason shows the way to go. No conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
Hey everyone, this is This Week in Science, and thank you so much for being a part of the show. Thanks for watching, thanks for listening, thanks for being there. If you are interested, you can head over to TWIS.org. TWIS.org is the portal to all things TWIS. We've got information there on recent episodes and show notes with links to the stories on every episode there. We've also got links to the audio files of the show. We've got announcements. And you know what? You can pre-order your 2018 Blair's Animal Corner This Week in Science calendar over there at twis.org. That's right. All you have to do, go on over there, click on the pre-order your 2018 calendar image. There's an image, TWIS 2018. Big bufo going on over there, some kind of toad. Blair loves her toads. Hypnotoad, but it's not a hypnotoad. You can order it and we will mail it to you before 2018. You'll get your calendar. You can start planning for next year. Pre-orders are open now. So head over to twis.org and get yourself a calendar. Come on. It's a coloring calendar. You're going to love it. We all love coloring. It's a coloring calendar. Additionally, over there at TWIS.org, you can also find other things that are pretty cool and TWIS related. Our Zazzle store link. If you click on that Zazzle store link, it takes you right to our Zazzle store, where there are multiple wonderful products that have the TWIS logo emblazoned all over them. Additionally, images from previous Blair's Animal Corner calendars, and I do think there's some good stuff in there. You can get a Tyrannosaurus Rex coffee mug, a tortoise mouse pad, a t-shirt with a toad on it, or a trucker hat with the TWIS logo. There's all sorts of cool things. I mean, really, seriously, that's just the first page. We have so many neat products. You can even get TWIS stamps for sending out your holiday cards. Come on, how can it get any better? Support TWIS while at the same time shouting your love of TWIS to the world. And then if you head back over to TWIS.org, you can also just support us, help us produce this show financially by clicking on the donate button. So go donate to TWIS right along the right sidebar, click on donate. It'll take you through a PayPal interface where you can donate however much you feel like giving to help us keep this show going. You can also click, if you want to donate and support us in an ongoing fashion, week after week, month after month, click on the Patreon link. That Patreon link will take you to our Patreon community page. Patreon.com/thisweekinscience is where that's located. You can become a patron and pledge to support us at whatever amount you feel like you can do per episode. So $1 an episode, $2 an episode. Each level that you support us at, there are cool little rewards related to it, and you get the inside track on things that are going on with TWIS. There are special emails and messages that come out for you, and sometimes you get previewing of videos that we're putting out when we put out videos. So being a part of the Patreon community and supporting us over there does have its perks. If you're not somebody who likes to buy things, and if you're not someone who is able to help us out financially and keep this show going in the producer way, we could really use your help getting the word out about TWIS. Seriously, tell your friends about TWIS. We could use the help. The more people we can reach, the better, right? The more people we can tell about science, the more conversations will be had about science on a daily basis. Sharing science is a great thing. It's like joy germs. They spread. Science is joy germs. Anyway, this is all a big request because, we do this show and we love doing it. We do it because of you, but we do it because we love it also, but we can't do it without you. We really can't do it without you. So thank you so much for your support. We can't do it without you.
Even bothering to stop and think. The only real power that I do believe is the dollars and cents all these authors receive. If miracle wonders were held in their looks, why waste precious time and try selling their books? Why sit and wait for your publishing royalty?
And we are back with more This Week in Science.
Yes, we are, Justin. What you got?
I got textiles. Textiles represent one of earliest human craft technologies and applied arts, and their production would have been one of the most resource and labor-consuming activities in the ancient past. So certainly something we would want to study about the past to give us insights into what people were up to back then. There's a little problem, though. Textiles are relatively rare finds in archaeology. Not just because people ran around naked all the time, though I'm sure that happened some places. But typically, conditions are unfavorable for something like organic material to be preserved. Many archaeological textile fragments do survive, however, in mineralized form, which forms the basis of a new study published in Antiquity. Not published a long time ago. There's a publication called Antiquity. Detailed analysis of several hundred textile fragments has provided for the first time a much more detailed definition of the textile related cultures in Italy and Greece during the first half of the first millennium BC, or about 3,000 years ago. According to Dr. Margarita Gleba, the study author and researcher at the University of Cambridge, luckily for us, during the Iron Age, which is 1000 to 400 BC, people were buried with a lot of metal goods, such as personal ornaments, weapons, and vessels. These metals are conducive to the preservation of textiles, as the metal effectively kills off microorganisms which would otherwise consume the organic materials, while at the same time metal salts create casts of textile fibers, thereby preserving textile microstructure. This is how we get such a large number of textiles, even though they only exist now in tiny fragments. Through meticulous analysis using digital scanning, through electron microscopy, high-performance liquid chromatography, and other advanced methods, we're able to determine a lot of information, including the nature of the raw materials, the structural features, such as thread diameter, twist direction, type of weaving or binding, and thread count. Technical differences suggest that during the Iron Age, textiles in Italy more closely resembled those found in Central Europe's early or Proto-Celtic cultures, while the textile culture of Greece was largely connected with the Near East. Dr. Gleb added, there is overwhelming evidence for frequent contact between Italy and Greece during the first half of the first millennium, about 3,000 years ago, but this evidence shows that their textile traditions were technically, aesthetically, and conceptually very different. This means that the populations in these two regions are making an active decision to clothe themselves in a certain way, and it may have to do with traditions set up already in the Bronze Age. And I found this story kind of interesting because we know that the next 1,000 years, or the 1,000 years forward, is going to start finding Romans and Celts going to war with each other, as though they are completely different peoples. And by Roman times, the establishment of Greek colonies in southern Italy, this is quotey voice still from the researcher, southern Italy and a more general Oriental influence observed in material culture of Italic populations leads towards gradual disappearance of the indigenous textile tradition. Our future research will attempt to understand the cause behind this change in textile culture. But it's very interesting because for a long time, because the Romans were so technologically advanced, a lot of things like swords and weapons and artifacts that are considered Celtic origin, and Celtic is sort of a large, that's from Iberia up through northern France out to Austria. Like it's really a broad region that is called these Celtic cultures because they shared a lot of tradition, a lot of technology, and there was a lot of trade in back and forth. But one of the items is this sort of iconic sword that's very, very Roman looking that they've found indications of within Celtic cultures dating back to the Iron Age. And so how curious now do we find Iron Age Italy having technological features similar with more northern Celtic, Proto-Celtic, early Celtic cultures. So yeah, and it's one of those things too. Again, that we look at a dominant culture and its advanced technology, and somehow throughout history have been assuming that that's where the barbarians of the Celtic North got their technologies from. And as we get more evidence, these technologies, it's sort of like the braided stream of human genetics, right? We find there's a braided stream of technology and culture and innovation that took place throughout these regions, from bronze into the iron and back into the Stone Age even. We were doing a whole lot of sharing.
Yeah, well, I think it's similar to, we know that the Greeks, as they took over various and conquered various areas, they did so not by implementing their own cultural standards over the people that they ruled. They said, okay, we're going to rule you, but you can still kind of do your own thing. And that allowed them to hold a much larger area because people didn't feel like they had to fight. They could still be themselves. And so by doing this, there was increased trade. There was increased influence between these different cultures as the empire grew.
Right. Yeah. I just found it large. I mean, this is yet another example, though, I think of the history of pre-Rome being. I tempted to even go so far as to call them potentially another Celtic, to fall into that another area that could have Celtic origins. Again, that term, very broad. It's pretty much become all of Europe before Rome, and has such similarities with what was going on in Scandinavia and the language and all this. But it's always fascinating that these things come about. We can find these microfibers through this wild transition of finally having that metal in there, preventing corrosion and creating these sort of salt casts of the textiles that we can suss out these details that would otherwise be lost through history.
And it's these little tiny details that are helping us tell a richer story.
Yeah.
And that is so cool. So neat. A couple of years ago, I reported on a discovery in mouse brains that the brain is influenced by the body's immune system. Historically, in physiology and anatomy, it was not really thought that the immune system of the body worked so much within the brain. It was like the brain has its own immune system. It's got its own cells up there that do things.
And we were protected, right? There's like a blood-brain barrier thing. It's like, no, the brain is a separate organ, not connected to the rest of your body.
Yes. But a couple of years ago, there was a discovery that lymphatic vessels, so our lymph system, our lymph nodes are where our immune cells are created and they're sent out to attack invaders in our body. And our lymph vessels are the vessels like blood vessels, but not. They carry lymph fluid, lots of immune cells and compounds. These vessels actually exist alongside blood vessels and get into the brain, into the dura mater of the brain.
Which as far as recent discoveries go, and as much slicing, dicing of the brain that has been done for all this time, to only just have recently discovered, oh, look, we some other little thingies here. We noticed before.
Yes. And so there is a new study just published in eLife, which is an open access journal, by researchers at the National Institute of Neurological Disorders and Stroke from the National Institute of Health. The researchers have used MRI technology, magnetic resonance imaging, and also cadavers to be able to discover lymph vessels in the human brain, not just in the mouse brain, but now in the human brain and also other primates, to give evidence that this wasn't a one-off thing in the mice. This is a cross-species mammalian trait that we have lymph vessels that get into the brain. And so our brains may drain out some of the metabolic waste of all the activity that occurs in the brain through the lymphatic vessels. The lymph system is also considered the body's sewer system. So get rid of stuff, pump it out through the lymph, right? The researchers, the head researcher for this study, Daniel Reich, said, we literally watched people's brains drain fluid into these vessels. And this was not something that was taught to him in medical school. And so to do it, they used MRI to scan the brains of healthy volunteers who had been injected with a compound called gadobutrol, which is a magnetic dye that's typically used to visualize brain blood vessels that are damaged by disease. The dye molecules are actually small enough to leak out of blood vessels in the dura, which is the kind of the membrane around the brain, but they're too big to pass through the blood-brain barrier to get into the important parts of the brain. And so when the researchers set the MRI to see blood vessels, the dura mater lit up really brightly because all of this dye was leaking all over the place, and they couldn't see anything to do with the lymphatic system. But then they took the scanner and they were like, okay, we have to do something so we can see this differently. And they tuned it so that the blood vessels disappeared. And when they did that, they found that the dura mater also contained smaller but equally bright spots and lines that they suspected were these lymph vessels. And so they suggest that the dye leaked out of the blood vessels and then flowed through the dura and into the neighboring lymphatic vessels. And so then they did a bunch of testing with different size molecule dyes, and they saw the blood vessels again, and then they did something else to see the lymph node, the lymph vessels again. And so they kept doing it over and over again, retuning their scanner and putting in different size dyes to be able to see the leakiness and where the dyes would go. And what the team says is really this is going to change the way we think about the brain and how it interacts with the immune system. And this is specifically important for neurodegenerative autoimmune disorders like multiple sclerosis, diseases in which the immune system is attacking itself and then attacks the nerve system. Understanding that there could be a disconnect between the blood vessels and the sewer system, that if you're not connecting correctly, then maybe you're not draining, and if you don't drain the sewer system, you end up in a swamp, and that can have all sorts of problems. So it is it's an interesting, a very interesting discovery that is exciting. And, this one, it was first exciting that it was found in mice a couple of years ago, but now that they have actually confirmed this is something in the human brain as well, that this lymphatic system is there, it could change the way that diseases are observed and also that they are treated.
Yeah, and again, this is wild. This is wildly fascinating, but it's sort of a refrain we've made a few times. Throw out all the other drug treatments and ways you've been targeting a disease, because now we may have found a direct thing that's being affected, regardless of the drugs you're implementing or where you've been witnessing symptoms. And for something that's affecting both the immune system and the brain, is it, well, we've noticed a correlation here, right? Pretty strong.
Now there's a connection.
Now we know that there's a way they're connected. Now we have an actual thing to target and look at and finding cures. This is huge.
Yeah. That the brain has a drainage system like the other organs of the body, and that we need to be looking at that. Need to be looking for it. And yes, clever trash panda, it is insane in the brain. Insane in the membrane. That's right.
Cypress Hill. Very nice.
Yeah. What do you have next, Justin?
Oh, is it me again? That was all of my stories. Oh, wait, no.
It never is.
Justin always trying for the fake out.
It is the most abundant element in the universe, a potential clean fuel of the future. Water and fossil fuels contain large amounts of it, but unlocking it from these sources takes a great deal of energy, casting doubt over any future. What's the element? Hydrogen economy. Turning water into hydrogen using solar energy could lead the way to cheap and clean hydrofuel. However, this process currently relies on photocatalysts containing expensive precious metals, limiting its capacity because of money and availability of these precious metals. Now, researchers centered at Osaka University have developed a new kind of photocatalyst for producing hydrogen from water. which is not only free of expensive metals, but also absorbs a wider range of sunlight than ever before. They've recently reported their findings in the Journal of the American Chemical Society. Quote voice, lead author Tetsuro Majima says, "We were pleased to find a good amount of hydrogen produced from water using our new composite photocatalyst with graphitic carbon nitride and black phosphorus. But what we didn't expect to find was that even when using low energy light in the near infrared, the photocatalyst continued to produce hydrogen." So this is potentially heading down into the future. Still more implementation. But the hydrogen economy's hugest obstacle right now is coming up with enough hydrogen, even though it's the most abundant thing in the universe, coming up with enough to use as fuel
cheaply and easily.
Yeah. But if this pans out, and if this can be scaled up and turned into hydrogen producing plants, we could turn 3/4 of the surface of the planet into this energy. Yeah, this would be a fantastic breakthrough.
Yeah, it would be an amazing breakthrough to be able to, right? The technology is what's holding us back, but we're making these incremental advancements. And so just fascinating. We have to take the elements we have, the things that are available, figure out what's abundant enough, that's cheap enough, that's easy enough to access. How can we get to it without, we want sustainability. How do we put it all together so that the next thing we create is a viable source?
Right. And it's one of these, this is sort of a fascinating thing that's sort of happened too, is that the hydrogen fuel cell car exists now. Yep. It's on the roads. It's in, it's at least in California and Oregon, I think New York. But it's on the road. There's fueling stations. People are enjoying the heck out of these vehicles that have zero emissions. Now we just need to produce enough fuel for them to keep driving these vehicles.
Yep. Make it accessible.
Without using a fossil fuel or heavily extracted energy somewhere else. Right. I mean, that's we got the vehicle. Now we got to clean up the fuel source, which is a lot of what's going on. I mean, somebody was joking that Chevron Corporation was opening up. This is a joke, by the way. This isn't a real thing. But they were opening up electric car fueling stations where you could go in and plug in your car. And that when you would plug in your car, though, you would hear off in the distance the diesel generator fire up to do the charging, which sort of would be defeating the purpose of the thing. Yeah. But yeah, we need to. The fuel source itself needs to have its own green footprint for this to be successful and meaningful.
Agreed. Yeah. Do you know what people have thought is meaningful?
Oh, plenty of things.
Lots of things. People get crazy ideas, actually. And one of those crazy ideas is that Tabby's star, this crazy, strangely dimming star that dims and brightens and dims and brightens. And what could be causing that dimming? People are like, oh, it's got to be aliens. It's a Dyson sphere. They're coming up with these really kind of off the wall suggestions, hypotheses, though, testable hypotheses, all as to what could possibly have been causing the dimming of Tabby's star. Now, I say hypotheses because the idea that it was possibly a Dyson sphere created by aliens was initially proposed by scientists themselves. Wow. Not just weird people coming up with ideas on the internet like we do. So KIC 8462852. This is Tabby's star, otherwise known as Boyajian's star, after Tabitha Boyajian, who discovered the star. And it does have these unusual brightness dips. NASA's Kepler Space Telescope observed it dimming over about 20% over just a matter of days, not a very long time. But there's also a longer term, there's a short-term trend, and there's also a longer-term dimming trend that occurs. But when stars are about the size of the sun or even a little bit more massive, we don't expect them to have this kind of dimming behavior. And so there have been all sorts of ideas that have been put forward. Well, there was a new study. Researchers started looking at it again, and NASA's Spitzer and Swift missions, as well as the Belgian Astrolab Iris Observatory, were taking a look at Tabby star and all the wavelengths of light that they were able to see. The study is published in the Astrophysical Journal. The researchers suspect it is not aliens. It's a dust cloud. Just a dust cloud.
But where did the dust come from, Kiki? Could
Well,
it be
that's
dust from alien origin? Okay.
Probably not. I mean, maybe an alien ship came by and pulverized all the planets around this star, creating a dust cloud.
And there we go. It's an alien mining operation that because you have to travel to deep space to find the elements that are kind of common everywhere in the
That's not what this study did not address where the dust came from. It was just looking at the light, and basically they found less dimming in the infrared light from the star than in the ultraviolet light frequencies of the spectrum. And so if there were larger than dust particles, any object that's larger would just be blocking light, right? And so then it's going to be dimming all the wavelengths of light. But if you've got dust particles, and of many different sizes, there's light that's going to be bouncing and scattering through the dust particles. It's going to be obscured and yes, dimmed, but kind of in a different way than if it's just big, bulky objects. And so the researchers looked at the way the light was scattered by the little objects. And so it suggests that the objects that caused the long period dimming of Tabby's star are only about 1/10,000th of an inch in diameter. Tiny little dust particles. Yeah. They observed it in ultraviolet using Swift, infrared using Spitzer, and in visible light using the Astrolab Iris, which is a public observatory near the Belgian village of Zillebeek. And the researchers, based on this strong ultraviolet dip, determined the blocking particles must be bigger than interstellar dust, small grains that could be located anywhere between Earth and the star. And these small particles could not remain in orbit around the star because pressure from its starlight would drive them farther into space. Dust that orbits a star, called circumstellar dust, is not so small that it would fly away, but also not big enough to uniformly block light in all wavelengths. So, little dust bits that could be around the star, but also might be anywhere between us and the star.
Okay, so alien dust. Got it.
Alien dust, right? The really fun thing about this is that the Astrolab team involved amateur astronomers. This was a citizen scientist project of sorts. These team members from Astrolab, they volunteer at the observatory. Frankie Dubois, he operated the telescope during the observations. He used to be the foreman at a seatbelt factory until he retired. Now he operates the telescope. Ludwig Logi, he helps with technical issues on the telescope, is a security coordinator in the construction industry. And Steve Rao, who processes observations of star brightness, is a trainer at a Belgian railway company. These are all just astronomical enthusiasts. Siegfried Van Verbeek, who's also a volunteer, holds a PhD in physics and got interested in Tabby star after reading an initial study and persuaded all these guys to use Astrolab to observe it. And they all decided that would be a lot of fun. And now they have published in this study on what's going on. So they have this good idea of the long-term dimming, but doesn't really explain that 20% dimming that was observed by Kepler. over about a 3-day period at one point in time. We really don't know why that happened. So this explains long-term dimming trends, the dust, but not necessarily short-term. Researchers do suggest that maybe it might be a swarm of comets that could be involved. There could be dust and comets that come through occasionally. So could all be related. But anyway, not aliens.
Not aliens.
It's just dusty in space. There you go. We've talked before on the show also about universal flu vaccines, right?
Mhm,
The
mhm.
idea that maybe there will be one shot to rule
Oh,
all
that
the
would be
flu
so
vaccines,
great.
right? It would be so cool. Well, it might be coming sooner rather than later. Fingers crossed for the first trial, the world first trial for a universal flu vaccine for individuals 65 years of age and older. They are going to be recruiting individuals in the UK. It's going to be supported by the National Institute for Health Research and will be delivered by the University of Oxford in Berkshire and Oxfordshire. Oxfordshire. They're trying to get 500 individuals. They're trying to get 500 individuals for the study. And they're hoping to see that this is going to work well. They've passed a safety study. So this is actually to see if it works.
Is this a one and done vaccine? So
Pretty much.
It
So
wouldn't be once a year anymore?
it would not be once a year anymore. No, because the reason we have to have a vaccine once a year currently is because of the different proteins that are involved. The way that the vaccines work is it's like a pin cushion almost. The virus is like a ball with these little pins that stick out of it. And those pins on the outside are little proteins. And those protein markers, they change and mutate all the time in the flu viruses. And so we formulate our vaccine to target create antibodies against those external proteins. But what's happened for the universal flu vaccine they're testing is they are creating an antibody against proteins that are on the inside of the virus that do not mutate and change as regularly and that are more common to all of the influenza viruses. And so usually we have influenza A and influenza B, and influenza A affects the largest number of deaths annually. And so this is affecting this particular universal vaccine will be targeted at influenza A central proteins. And so hopefully it will protect people against influenza A.
That would be great.
Yes. And so they are going to be the initial trials. They are going to receive the regular annual immunization. So look at the regular flu shot so that they don't get sick if the universal vaccine doesn't work. And it'll be in combination with either the new vaccine or a placebo.
How will they know if it's working if they're also giving them the normal flu shot?
Well, they will be first testing to see they'll test people's blood on follow-up to see what antibodies their bodies are creating.
Cool. Very neat.
Yeah, it is very neat. And so we will see. And this is primarily aimed at helping individuals that are to be universal vaccine for aged 65 and over, as current vaccines are only effective in 30 to 40% of over 65s because of the weakening of our immune systems as we age. And elderly do suffer the most in terms of mortality from the flu as well.
Well, as soon as everyone's super vulnerable is taken care of, I'm ready.
Yeah, that's right. Let's do it.
Sign me up.
Yeah. So if you're in the UK, in Berkshire or Oxfordshire, Oxfordshire, then and you're over 65, this may be something to keep an eye on. Blair, tell me about bullies. Or at least
killer scientists.
Killer scientists. Yeah. What? No,
Yes.
Scientists are good people.
Let's
not
talk about it.
killers.
So some UK scientists have taken to an editorial begging for people to stop bullying them on the internet, calling them killers. That is because of a citizen science program that was started to get the beat on some social wasps in something called the Big Wasp Survey. Say that 5 times fast. And so how do you survey wasp populations? You catch them and you pin them and you compare and contrast them to categorize them. This is
I was gonna say just go to any cheesecake factory, but
right. So this is how entomology works. We can do a certain amount of citizen science with taking pictures and stuff like that, but ultimately, at some point, you got to pin some specimens to do a categorization of these individuals. So they wanted to find out about these social wasps. They're pest controllers, they're pollinators, they're really important. So they asked the public to set out beer traps, which is exactly what it sounds like. It's a water bottle you cut in half, you insert it upside down, and you pour a little beer in there. So the wasps are attracted to the beer, they get stuck in the trap. And then they were able to submit these wasps in the beer traps to the scientists, and they were able to start the survey. But in their ask to start the survey, public opinion exploded, calling these scientists killers and murderers, killing in the name of science, and lots of hyperbolistic kind of comments about what scientists do. So I just wanted to mention it because we talk on the show a lot about specimens in museums that have suddenly created these breakthroughs in saving endangered species, in discovering once lost species. And we have also talked a lot about pollinators and effects on pollinator communities from climate change and all sorts of things like that. How do we know anything about that? It's from scientific studies that involved capturing and pinning these individuals. So yes, perhaps someday you'll have an advanced app on your phone where you can take a picture of a wasp and it'll do a 3D model of it and send it to a scientist somewhere, and we won't need to capture or kill any animals. But as it stands now, these are the best methods that we have to conduct science. And I also want to mention that people in general are not that nice to a lot of insects,
Especially wasps.
especially wasps. So I do also want to call out kind of the hypocrisy in this.
Yeah, please.
Of using a fly swatter on an insect 1 second and then the next second taking to the internet in droves, calling scientists murderers for trying to study a species.
that's why this sounds kind of like a setup to me. Like, is there really that big of a pro-wasp community out there that this really like galvanized people?
I bet if you asked any of these kind of troller type people out there to come see a wasp alive up close, they would probably say no. Say no thanks, get that wasp out of my car. So it's an interesting conversation that I just wanted to bring up because it seemed unjust to me. But I think it's also important to remember why we do these studies. They are hugely impactful on all other fields of science in a lot of different ways. So yes, we wish we didn't have to kill individuals for scientific studies, but sometimes it's necessary to do proper science. And that science, that one individual, their sacrifice to science can make a huge difference down the road. So I'm not saying go out there and kill wasps. But what I am saying is if the scientific community, especially the entomological community, a community I'm very excited to talk to soon, who is extremely passionate about the work that they do, and they care a lot about their invertebrates that they study, that they would not have any unnecessary loss of life. and that really they're trying to save species by doing this research. So
right, the person who studies cockroaches is less likely to step on them than you are.
Yeah,
Probably
absolutely.
get down on the ground and observe for a little while.
Yeah.
Yeah, I wonder how many of these individuals who are up in arms over this have never killed ants, never put out a fruit fly trap in their own kitchen, never, never swatted a fly, never, let he who does not live in a glass house cast the first stone, right?
Yes,
It's, the whole situation. The whole situation. And yes, unfortunately, we are not at the point where we can do the science without the death of the insect. But it's
take some perspective and realize that it's part of this grand scheme to actually save species.
Yeah, and in order to save species, sometimes you have to do a little killing. Currently,
in order to make an omelet, you got to break a few eggs.
Currently,
oh my goodness.
1 million birds a day are dying due to a killer that is plaguing Australia. That's 377 million birds a year by this estimate, killing 338 bird types, almost half of Australia's native species, including 71 threatened species, such as the spotted quail thrush, the squatter pigeon, and the night parrot. The culprit? An invasive species, including and actually exclusively limited to cats. With the staggering slaughter driving down the decline of many species, a study published in the journal Biological Conversation estimated that wild cats wiped out 316 million birds every year, while pet cats killed 61 million a year. Everyone knows that cats kill birds, in quotey voice, but this study shows that at a national level, the amount of predation is staggering, says lead researcher John Wojnarski from Charles Darwin University. It is likely to be driving the ongoing decline of many species. Numbers are based on results of nearly 100 studies across the country by environmental scientists, each sampling cat population and density. Another set of almost 100 studies assessed feline diet. Feral cats, which number in the millions across the country, considered the main culprit behind the high rate of mammal extinction in that country as well. They have wiped out entire populations since being introduced by Europeans, with efforts to cull or sterilize them so far failing to slow the march. We found that the birds most likely to be killed by cats are medium-sized birds, birds that nest and feed on the ground, birds that occur on islands or in woodlands, grasslands, and shrublands, said Wojnarski. For Australian birds, cats are a long-standing, broad-scale, deeply entrenched problem that needs to be tackled more effectively. Australia's Threatened Species Commissioner, Sebastian Lang, called the study extremely important and of great concern. Our knowledge of the impacts of cats on threatened mammals was a major stimulus for our first ever national threatened species strategy, which prioritized actions of controlling feral cats, said Lang. Under the government's threatened species strategy, more than 30 million Australian dollars, which I don't know, that's probably like 400 American dollars, has been mobilized for projects to reduce the impact of feral cats on wildlife. In June, construction began on a huge cat-free zone in the desert, covering 69,000 hectares. That's a meaningless word. 170,500 acres to allow native mammals that have been pushed to the brink of extinction by the predator to be reintroduced. So big thumbs up to Australia for their cat culling activities and for creating, which I think is a brilliant idea, huge cat-free zones. I think we need those across the world, really, to preserve our native bird populations from these invasive species.
Feral cats specifically are the big.
No, that's pet cats too. Pet
cats too. But the main impact were these feral cats.
And the secondary impact was the pet cats. And where do you get feral cats from? You get feral cats from pet cats. That's how you
Yes.
get them.
So fix your
That's where they
animals,
come
people.
from.
There we go.
Cull them.
Fix your animals. Fix your animals. Oh, and that brings us to another hot end of This Week in Science. We have done another show. Thanks, you guys, for a good show. Appreciate it.
Fun.
It was fun. Thanks, everybody, for watching. Thank you, all of you in the chat room, for your fun commentary. I love peeking over and seeing what you have to say in the chat room. I try and peek over at Facebook and YouTube as well. Thank you so much over there. And thank you to Fada, to Identity 4, and to Brandon for all their help in making TWIS possible this week and every week. I'd like now to thank our Patreon sponsors. Thank you, Columbo Ahmed, Tyler Harrison, David Simmerly, Marjorie, Aaron Luthen, Adam Mishkon, Craig Porter, Douglas Campbell, Joe Wheeler, Luma Lamed, Alec Doty, James, Jason Olds, Jim Drapeau, Mountain Sloth, The Honey Moss, Stefan Insom, Kurt Larson, James Dobson, Philip Shane, Emma Grenier, Matt Sutter, Salgid Sam, Braxton Howard, Phil Deneau, Greg S., Rick Ramus, Steve Mashinsky, Dave Wilkinson, Shu Wada, Artyom, Christopher Dreyer, John Crocker, Flying Out, Mitch Neves, Hexator, Mark Orley, Radio, Brian Hone, Felix Alvarez, Rudy Garcia, Darwin Hannon, Ben Rothig, Arlene Moss, Todd Northcutt, Dale Bryant, Brendan Minish, David, Paul Stanton, Dana Pearson, Christopher Rappin, Stir Frankadelic, John Atwood, Ken Hayes, More Cowbell, Steve Lessman, Robert Aston, David Wiley, Robert Rodney, Nathan Greco, Richard Porter, Jason Roberts, Eric Knapp, Matthew Litwin, Jason Dozier, Dave Neighbor, Jason Schneiderman, Alex Wilson, Haroon Sarang, Dale Lambert, XV, Timed Upper 319, Greg Guthman, Paul Harden, Louis Smith, Kyle Washington, Bill Kersey, Brian Condren, Steve DeBell, Gerald Unyago, Tony Steele, Randy Mizuko, Larry Garcia, Marshall Clark, Layla, Trainer 84, Ed Dyer, Mark Mazzaros, Bob Calder, James Rundle, Dave Friedel, Ulysses Adkins, Kevin Railsback, Steve Bickle, John Gridley, Brian Hedrick, Charlene Davidson Henry, Richard, Gerald Sorrells, Howard Tan, Jake Jones, Keith Corsell, Andy Groh, Tyrone Fong, Jacqueline Boyster, Eric Wolf, Daniel Garcia, Kevin Parachan, E.O., Byron Lee, Richard Onomus, John Ratnaswamy, G. Burton Lattimore, and Paul Disney.
The list is growing.
It is.
Thank you all.
Thank you for all your support on Patreon. And if you are interested in supporting us on Patreon, you can find more information at twis.org or just go directly to patreon.com/thisweekinscience. Remember that you can just help us out by telling your friends about TWIS. Tell someone about TWIS today. That will help us. On next week's show, we'll be back once again with more great science news. Lots and lots and lots of things to talk about. Broadcasting live on 9 at 8 p.m. Pacific time on Wednesday. You can join our chat room at twis.org/live. But if you can't make that, don't worry, because we have past episodes at twis.org/YouTube, at facebook.com/thisweekinscience, or just at YouTube or twis.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile type device, you can look for TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science and anything Apple marketplacey.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org, where you can also make comments and start conversations with the hosts and other listeners.
Yeah, or you can just contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. There's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We will be back here next week. And we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
It's this week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And this week in science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say. And if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.
Have my finger on that stop button. Stop it. Stop the music. Stop the music. We're in the after show, everybody. We made it to the after show. You know what that means? This is after the show. Not before the show, but after the show. We will be in Denver, Colorado in 1 month. Blair, you're muted. You muted on purpose?
Sorry.
Oh, there you are. Sorry.
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 One of my favorite characters from Saturday Night Live. Sorry.
Gilly.
Sorry. Gilly. Sorry. Sorry. Yes. Was during the show Ben Rothig.
What? What happened?
Nothing. Ben was being silly. Yeah, the after show kind of sometimes is during the show nowadays, but just kind of do the faux show.
What?
In a month. We're going to Denver, Colorado.
Oh, yeah.
1 month from tomorrow. Yes?
Yeah.
Yes. We'll be in Denver, Colorado. Anyone in Denver, Colorado? I hope that we can meet up with people. That would be fun. So I don't know where Dave Friedel is.
Last I checked, he was in Denver.
He's in Denver, right?
But he's not in the chat.
He's not in the chat. So we can't chat and say, Dave, coming to Denver. He's going to have to listen to the show later. Who else is in Denver? I don't know. We'll find out. I guess I have to start telling people that we'll be in Denver. More than just this right now. Pack jackets. I know there could be weather. What's in Denver? The Entomological Association. Entomological Society of America. That's right.
Yes.
You know, all those killers that are killing wasps.
Oh yeah, the wasp people.
Right. Wasp killers.
The etymology one. Is that also in November? Or is that a different?
No, that's different.
That's a different thing.
The what one?
Thing. Oh, Portland podcast. Well, somebody is throwing a podcast festival in December here. I guess I'll find out about it.
Whoa.
But it's like pretty fast after Denver. It's kind of crazy.
When in December?
I don't know. Nobody's invited me, so.
Oh.
Yeah. Some group called Fun Employment. What is this?
Yeah, never respond to those ads.
No.
Never apply to that job.
Yeah. Fun Employment Radio.
Yeah.
Wow, they've been podcasting 5 days a week since 2009. I don't even know who they are. Hence why they didn't invite me. Who's Jason Lamb? Who's Greg Nibbler? Who's Sarah X. Dillon? Do I need to know these people? Ooh, Unemployed Festival. Unemployed for a week. That's okay, Ben. Fun Employee. Oh, I know it's a well-known podcast. I just haven't listened to it. I will admit. I know it's been around for a while. I didn't realize they were in Portland. They're in Portland. And it's going to be just down the street from me at the Hawthorne Theater.
Yeah, look at that.
Yeah, maybe I should get in touch with them.
Yeah.
Say, hi.
I'm your neighbor.
I'm your neighbor. That's right. Maybe I should find out. Yeah, I'll go. It's right down the street. Why is everything so hard to find out about?
The interwebs are just too big. That's why.
The interwebs are a very big place. It is very true. There's a link to that. Oh, they've got a Facebook page. There we go. Here we go. PDX Pod Festival. Saturday, December 2nd, 5:30 p.m. Shows begin at 6.
I'll be right back, friends.
Maybe I should find out who they are.
One moment.
Okay, friend. Come back. These names are familiar. Who are these people? Yeah, I know who these people are. Anyway. A TWIS minion. Yes, therapeutic quality of homebrew. That could be a good topic. I know, I can't listen to them all. That's right. Clever Trash Panda. Yes. We'll go to the Hawthorne. We'll go to a podcast festival. Yes. That'd be fantastic. That'd be fun. So let's see. People are still talking about kittens and fixing their kittens. And then Ben Rothig's taking off time between jobs. You should have taken 2 weeks off. It's nice to have some time off, but you're not making money, so that's another problem. And then we've got people talking about Australia because Australia. And yes, I think the pod festival is for PDX pod people. I think so. We say feral cats, right?
Feral, I thought.
Feral.
Feral.
Could be feral.
Feral.
Did I mispronounce it? I don't know. It's getting late. I'm tired. I haven't had any alcohol, unlike other people who are watching the show.
Or on the show.
I'm trying to buy a microphone stand, meanwhile.
Oh, good.
Let's see. I don't know the difference between these doohickeys.
I had a... What? Oh, you just need something? What do you want? Something that's on the floor.
Yeah.
And it has an articulating arm, right?
Yeah. There's a bunch of them. I don't know what to choose.
You don't need a...
There's so many objects.
It's a mic stand. You can do this, Blair. Did I find out what the problem with Patreon was? No, I don't know what happened.
Wait, what? There's a problem?
Yeah, so September, somehow, I mean, I possibly could have not posted the last show of the month in Patreon, but I remember sitting down and going, oh, I forgot to post the shows in Patreon, and I have to post the shows in Patreon for people to be charged for them.
Right.
And I started looking, and or Brian Condren contacted me and said, I haven't been charged for you guys. I've been charged for all the other podcasts and other things that I support.
Weird.
I haven't been charged for you guys. And I was like, that's really weird. And he said, yeah, that's really weird. And so then I went into Patreon and discovered that none of the episodes that I had posted actually posted. And I don't know why. So I did put them in as scheduled posts. And so I'm thinking that the whatever mechanism, either I didn't hit a button to I might have done the scheduled post and then not saved it or something. It's probably my error. But yeah, that's not going to happen again. So anyway, nobody got charged. We didn't get any money this month or last month. And but then on Monday, I went through and I put all of the episodes from September in. So now people who are on Patreon may be charged for lots of episodes this month unless they have limits on how much they can. There's limits to their support. A top limit on Patreon. So yeah.
Phooey.
Huh.
Yeah, it'll be fine. It's all good. We survive.
Okay. I've narrowed it down to 2.
Sweet. Show us. Strengths. I love that image. That's hilarious. Strength says it would be funny if I found out I'm in a neighborhood packed with famous podcasters and they're all operating from their garages. I think that would be pretty funny. Nobody knows anybody else is here. Everybody's in their garages.
Do a podcast garage crawl.
Oh, that would be funny. That would be a lot of fun, actually. Yeah. Oh my goodness.
Be guests on each other's podcasts. Just hop around.
Yeah. Let's see. Also, YouTube has changed rules around videos with external links. Yeah. They don't like you they want you to use their platform. And they don't want you linking out to Patreon and all that kind of stuff anymore. So I watched a video recently. YouTube has kind of like this inside the algorithm video series, podcast series. And I checked out one episode, and one of the engineers was the head engineer was talking about the YouTube algorithm, and one of the questions asked was, okay, everyone wants to know, how do you get more views? And the engineer gave an example, and he said, well, we wanted to try something first. We looked at the views that different videos were getting across YouTube, and we found that there was, for engagement, that there were some videos where people were watching for an hour and a half. They were watching and not turning it off. And I was like, yeah, that's my audience. Yes. And then the engineer goes on and says, but there are a very small minority of YouTube's viewers. And so while YouTube does want to support those viewers and their needs, really YouTube is focused on growth. And so we changed the algorithm to exclude videos that are long. So basically all of TWIS's videos are much too long for the YouTube algorithm to promote, because YouTube is focused on getting new viewers, and the maximum number of new viewers will come if videos are shorter, because that's what their data suggests, is that people want short videos. So if you're not a short video, their algorithm will not prioritize you. Ta-da! So now I've discovered that we need to do what I've decided we need to do as a result of this is have a preview video where I'll go on for like 5 minutes, or not even 5 minutes, 2 minutes, or Blair can go on for 2 minutes, or you, Justin, can go on for 2 minutes and summarize, say, hey, don't miss this week's episode of This Week in Science. We're going to be talking about, and give a summary of the stories in the show. It'll be like a quick summary. Be like, This Week in Science is live every Wednesday. Don't miss the show tonight. See you later. Bye. And then we've got a short video, and that video will get promoted. And then I can put these cards I can put things all in the videos so that people will click on the long video and hopefully watch it. This is my YouTube strategy, if I care about YouTube and doing that. Yeah. This is what I need to do for the YouTubes.
Well, I don't know. Would
What?
like the YouTube is a conduit for us to reach the audience we've kind of already got, largely. And yes, we want growth. We want more new. I don't know though that's the new like if you are just clicking into that, is that the thing that you're like, oh, I'm going to watch this thing that's a commercial for another show? I don't know that I'd even want to watch it. But then you already are watching it before you know you're watching it. Like, I don't really understand how it works. Yeah, I don't know.
M-Beam says, just switch to Twitch. It's probably the solution. That's probably the solution. Twitch is a little bit nicer to its partners. Yeah.
Would that mean we're not on the YouTube, or does that mean we're going through Twitch and then it sends it to YouTube later? Like,
yeah, that would kind of be. It would either do that, or if you have, there's if you have a publishing software like OBD, Open Broadcast Distribution, OBD, whatever it is, Open Broadcast, OBS, Open Broadcast System. If you have a publishing software set up, you can use it to publish to multiple places at once. And so it can send, if you have the addresses to send it to, you just input those, and then it'll send, you do it once and it sends the feed. But because we're using Google Hangouts, we couldn't necessarily, I don't know how to do that. I think we would have to switch how we do what we do. Things would have to change significantly. Google Hangouts is super easy. That doesn't mean it can't happen. It just means that I would need to make things happen.
Blair, I like your more expensive mic stand better.
You like the $60 one?
No, it's 60.
Okay, because
No,
Bleak,
cheaper on
Bleak,
mine.
I
Mine's
know.
34.
Bleak just
Or
sent
35,
one that's 60, but it seems like overkill. It's very nice.
The one that says here's a gem of a mic stand. Whenever anybody, this is like sales lingo, I'm a little bit behind the scenes. Whenever anybody's selling you anything as a gem of anything, it's because
Where does it say that?
It's because it's crap.
Where does it say here's a gem of a mic stand?
On an oh-hoo-hoo microphone stand?
Hoo-hoo.
Whatever. The first thing in its description. Here's a gem of a mic stand.
Okay, so hoo-hoo's out.
Junk.
So now
Yeah,
I'm looking at the Lix Pro, and then I'm looking at the pile that Bleak sent. But that one is $60. I don't know.
I like the Lix Pro. I think that one's just fine.
Yeah. Tree new B microphone. Come on, open. What's that?
That's the
No, that one goes
mount
desk
version.
mount. You
Yeah,
don't
so
want
I don't
desk
want
mount.
that. Mm-mm.
Yeah.
It's the one below that one.
That's what I've got.
Yeah.
Thanks
The
to-
pile is pretty good.
Yeah, it's $60 though. I mean, it's nice because it has like the springy part, so I wouldn't be stripping all these screws, which is what just happened. I stripped like 3 screws over the life of this thing, but
yeah,
I don't know. Kiki, it's your call.
Actually, that's kind of nice. Okay, it booms out a little bit, which is convenient.
Yeah.
You're gonna this
which could mean also booming over.
Mm-hmm.
It's like an old table lamp design.
Reviews.
Yeah.
Good for the Yeti if you have a counterweight.
Yeah, will the snowball work in this? That's the other question.
Solid and secure.
Yeah, I think it would just hang down upside downy. In face for it to be fine. But I think again the Lix Pro would probably be fine too.
Yeah, everybody says it's a decent stand for the money.
Hey, there's only 2 left in stock. You should order now.
Other people say the weight of the mic pulls it down and they have to keep retightening.
That's not great.
Yeah. The pile probably will not have that problem, at least not as soon.
Okay, you think I should go for that one? The one that looks like a lamp?
Yep.
What are the votes? What are the votes? What does everyone vote? Ooh, Restream IO. Thanks, MBeam. Look at that. Oh, look at that.
Strengths. How did Jacksonfly fit? I don't really understand how it works into one syllable.
I don't even podcast, and I kind of want one.
Practice. That's how. Practice.
Suspension scissors, sound dampening product. We do want sound dampening for sure. And the way that you would set it up, could we get it so it doesn't hang in front of your? Well, I mean, it'll kind of hang in front of your face, like.
Oh no, Gem Doctor! I didn't mean any disrespect by.
What did you
the
say?
Gem of something, meaning it's junk. You obviously are the real thing that they're trying to compare their junk products to.
Ben Rothig says pile is aptly named.
Oh no.
That's funny.
Yeah. So
Vote
I don't
for
know.
the one with Amazon Prime, says Ed.
Right.
Oh yeah, that's not this. Is this one Prime? Pile is prime.
The Lix Pro isn't, but
Lix Pro is not.
I need it in a week. I feel like that would be fine.
Yeah.
So
the table
Okay, so
mic
just-
stand Chris Clark got me is absolutely amazing, by the way. This thing has not let me down in any way.
yeah, unfortunately I need a floor stand.
No, you just need a desk and a mic stand. That can not do it.
I don't do desk.
You need to start. This is what
No,
revolution is.
when I sit for the show, I fall asleep.
Wow.
So glad we
Wow.
can keep you awake.
Maybe not.
All
one of us
right,
after all.
okay. No, I'm just less my muscles are less activated. I'm better at like all my blood moves when I stand. It's better. Everything's better about it. Kiki knows she's doing it right now.
What?
Standing?
I'm standing. I
Yeah.
know.
Standing is the way to go. Active body language.
I would like you to have a microphone. Do you want one that, I mean, if you have the pile, it will hang down in front of your face.
Yeah, that's probably not great. Will it? Or can I scissor it down? Oh, yeah, I guess it will.
Well, actually, no, because of the way that the, let's see, let me look at that picture.
Because I should be able to make it come from the side. In theory.
Or even underneath your face.
Yeah.
Yeah. I just want to move that pop screen.
But it has that back sort of space to the elbow, which might get in the way of where you're trying to put it.
Yeah.
You might be better off with something straight up and down.
Mm-hmm.
Which apparently it can also do, but then I can't tell how low.
Yeah, I'm trying to look at it. 28.3. No, that can't be right. What tall is
Mic
it?
arm extension distance 3.1 feet. 3 foot 1 inch. Doesn't say how tall it is though. Where are the specs? Show me the specs, man.
All right, here's what you do. You buy the cheaper Lynx Pro and a sandbag, just so you can weight it.
Oh,
If need be.
Yikes.
And some duct tape.
Trying to ditch the duct tape.
Oh no. Now you've sent me to a Amazon sort of generally specific audio.
Uh-oh.
Now I'm gone.
Now you're in
I'm
trouble.
oh, danger.
Justin found
No,
toys.
stop. I have too many electric guitars as it is for somebody who's not in a band or even proficient with instruments. Stop. Leave me alone. Leave me.
Because you bought a previous electric guitar. How about this electric guitar?
Oh my gosh. Actually, that's one thing I haven't bought from Amazon. I don't think any of that came from it.
Then it's just been listening to your phone.
Yeah.
Look at
Oh, no
these
doubt.
cool
They're all connected
things.
now anyway.
They've got mic and tablet stands, so you could have a tablet that you read from as you're talking into your mic. Look at
Well, I've
that.
seen those.
That's clever.
Oh, it's clever.
Let's see what's going on in deals. Just give me something that's a deal.
I want sponsored products. I want to know what people want. And a microphone stand. Come on, people. No, not cables. What are some other microphone stands?
Now I want to go play guitar. This is going to be it's kind of loud at night.
Yeah, I found one, the Flex Zion tripod microphone stand with an extending boom arm.
Okay.
It's $25.
Okay.
It's Amazon Prime.
Okay.
Okay. All right.
I don't know about these things.
Okay.
Okie doke.
Everyone's like, I don't know anything about mic stands.
Yeah, I definitely don't know anything about mic stands.
Why are they Pokemoning us all of a sudden? What are we catching all? Guitar Mon. You guys are funny.
Flexion.
Wait. I want this marching percussion thing that you like wear. So you drums are in front of you and you like it harnesses over your shoulders.
Oh yeah.
And you
I was
could do
looking
that
at
with a mic stand. Replace the drums with the mic stand, and then just anywhere you turn,
Good.
it's like a baby Bjorn for your microphone.
So, Kiki, what's this clip for? That's one of the things I was really confused about. I did look at this one before. I was like, what
What
is this
clip?
clip on the Flexion? Why
What
is
do
there a
they do?
clip
What clip?
on the
Put the
on the-
microphone. The clip on the top of the stand there.
Yeah. What's that for?
That's a great.
That's when you
question.
slip in a traditional microphone.
No, not that. That's the traditional microphone, but there's
Oh.
it's a dual mic holder clip.
Yeah, what does that mean?
You could put another microphone in it.
You could put another boom arm.
You could put a
in it.
boom arm in it. You could put a
Interesting.
And then you could do duets
Good.
with one mic stand.
It's perfect for church.
Okay. Is that what someone wrote?
Yeah.
Rotating, folding for conference church studio audio DJ word salad.
Okay. Yeah, I don't know. I mean, I'll back your play. I don't know. If you think this is the
There's
one,
a-
I'm
There's
on
a
it.
Samson for $26. Newer.
I got one on the Prime here for 20. But
It's a gem of a mic stand, I heard.
Amazon Basics tripod boom microphone stand. Yeah, that one's $19.
Here's the Audi microphone
Probably
stand.
Tripped in a week.
Yeah.
It has 4 stars, but I don't know, what does that mean for, what you expect from a $20 mic stand?
Who knows?
I'm telling you that Lix Pro is 4.5 stars with 127 reviews.
Mm-hmm.
It's looking good. Looking real good.
Yeah.
Yeah.
That might be
Maybe I will just do that one.
Do it.
Okay. I knew.
Yeah, it's like what, $36?
$36 free shipping. Not Prime, but that's okay.
It's okay.
It's okay.
It's all right. It's okay.
Shipping to
Hi.
my address.
That's right, Whiskey Renegade. We'll put 10 mics all right in front of Blair.
Good.
Mm-hmm.
If there's one thing I need more of, it's amplification.
USS Rover wants to see your current microphone, but he doesn't know that if Blair moves her current microphone, everything will fall apart.
That is true. Oh my goodness. I'm going to get it on October 18th.
Not next week.
No. That's okay. I'll just limp along until
Can
it
you
arrives.
can you limp for another week?
Yeah, I might duct tape the heck out of this thing in the meantime.
You and the duct tape.
Yeah.
This
I
has
mean, if
happened
it's going to be
before.
trash anyway.
That's right.
Okay, order has been placed. October
Sweet.
18th to October 26th. We did a thing.
We did a thing. That's awesome.
Okay.
Something.
Oh my gosh.
That looks great, Schnuggo. I have some of those too.
I
They go bad.
have something I want for Christmas now.
This is the one that I have that just broke. But
On stage.
yeah,
Yeah.
that's the one.
Yep.
I just moved it around one too many times, I think. There's too many moving parts. But that's exactly what I have right here.
Yeah, it'll happen again. We'll need to get you a new one. These things, this is what happens. This is what happens with them.
Yes.
Screws get stripped. Wow, restream.io, the best way to stream to 30 plus password platforms simultaneously. Wow.
Oh, you can get a Theremin for $300 now.
It's a mini. I want it. It looks awesome.
That's really cool.
What'd you find? What you want for Christmas?
Theramini.
For Twistmas? Ooh, Theramini. That's cute.
Yeah.
Very fancy looking. Not like the old school Theramins.
That theremin looks like how theremins sound.
I think it looks like an answering machine.
What's an answering machine? What are you talking about?
It's something people use at the same time that people also use beepers.
I remember having an answering machine.
I had to explain beepers to a teenager recently.
Oh my god.
This is the mic I have now, USS Rover. Here I have
Snowball!
to be careful because it's falling apart. But
Snowball!
there it is. And then, oh, yep, there it goes.
That's just to hold the microphone for the rest of the evening.
See what just happened? Because I moved it. So I'll just do the show like this now.
There you go. Now you just hold it right in your face. In your face. I'm yawning.
Okay, is it bedtime?
Mm-hmm.
Let's have
Maybe it is bedtime. Yes. I'll find out about this podcast festival here in Portland. We will tell people we'll be in Denver in the beginning of November.
Denver in November.
Denver in November. And is there anything else in the next week that's going on?
Nope.
It's Statistics Day on Friday. People need to pre-order their calendars.
Yep.
And yeah.
Yep. Pre-order calendars.
Right then. I will see the two of you and read stuff that the chat room is saying again next week.
Absolutely.
Hooray!
Hooray. Thank you, everybody.
Oh my
Okay.
God, that reminded me, I held a chicken today, guys.
I like chickens.
That's great news. Now say goodnight, Blair.
Hold on, let me show you a picture of me holding a chicken.
I want to see this. I love chickens.
Why do you guys always drag out the end of that?
So I don't know how well you can see. Here's me and my two interns.
Why are you holding chickens?
Because I work at the zoo.
When you work at a zoo, you hold chickens. Okay.
And should I even ask? Do I even want to know what those chickens were fed to?
They are education chickens. They won't be fed to anyone.
Education chicken.
Here,
That is
turn this off.
It's a special denomination
There
of
we go.
chickens. Hold on, let me make you. There you go. There's your chicken.
I'm with a chicken.
It's a dark-haired beauty, just like you.
Yeah.
Okay, Blair.
It's an
Next time you
Australorps.
Next time you share, I'm just gonna throw this out. Next time you share a photo of you with an animal at the zoo,
Yeah.
a chicken.
It was fun. The chicken, it sat in my arms and it was very
Okay.
content, but it went.
So from where I live, there's where I live, there's people have chickens in their backyard. There's free range turkeys all around.
But it
So I guess
was very
somebody
like
in
a
a big city, even somebody in a big city who works at a zoo, coming into contact with a chicken is an amazing animal encounter.
It was fun, but it was definitely, I felt like the chicken was going, what's going on? You gonna put me down maybe? Are you're just, no, you're gonna keep holding me still? Yeah? Okay. What's up? Why am I off the ground? Why aren't my feet touching the ground? That's what it felt like. All right. And on that note, I will say goodnight, Blair. Say goodnight, Justin.
Good night, Justin.
Good night, Kiki.
Good night, Kiki.
Yay! Good night, everybody. Thank you once again for watching. We hope that you have a wonderful, wonderful week. Send us your pictures with chickens.